Okay, hello again, everyone. So last time we were just going through the fundamentals of biology, and this time we are picking up where we left off last time. So we are going to continue with cell division for today. What is cell division, the different types of cell division, and what's the purpose of it, as well as after that we will just go through a bit more fundamentals that you need to know about in biology. Typically, what happens inside of cells. There's like three crucial processes that you need to know about, but that's what we're going to be covering for today. And with that, let's get started. So, firstly, with cell division, okay, which is a bit funny because you can call it cell division, but really it's when cells multiply. So it's kind of funny that division is the opposite of multiplication. Yet you can use the same name for both of them. Either way, in biology, we tend to call it cell division. So in cell division, this is the process by which a cell will divide from one of itself into two of itself. Okay. Um, for bacteria, when this occurs, we call it binary fission. But in our cells, like eukaryotic cells and prokaryotic cells, we have or rather eukaryotic cells, because prokaryotic cells are the simpler ones of bacteria. So in eukaryotic cells, we instead call it mitosis or meiosis, depending on the function and the need, right? So we're just going to go through both of them really briefly. But before we can go through both of them, you need to understand something important about the cells. So if you remember with the eukaryotic cells, I'm just going to draw a typical animal cell here. Okay, but with every kind of cell that we have, right? Typically, those cells have a nucleus in the center. Okay, and that nucleus, which is not exactly that large, but that nucleus will be containing the cell's genetic material. Okay, typically speaking, this genetic material is spread out throughout the cell or throughout the nucleus, so it's a bit hard to see under a microscope. The only exception is when the DNA that's in the genetic material actually condenses into these smaller packets that we call chromosomes, right? And the reason why the cell does this is because when it's close to being ready to divide, it condenses the DNA so that it's easy to separate that DNA between both of the cells. The cell needs to pass on its DNA, otherwise the identity of that cell is basically lost, right? So in order to prevent the cell from going haywire, that DNA needs to be passed on, okay? Now, that leads us anyways to what I was talking about earlier, that when the DNA condenses, so when I have this spaghetti-like structure of DNA that condenses, we call this a chromosome. The most famous depiction of a chromosome is this butterfly kind of shape, where you have the DNA, that's this double helix structure, that's what DNA looks like, and it condenses and coils and coils and coils until it makes a structure that looks more like this. Okay, so it's a bit of a butterfly shape. Of course, in reality, it actually varies. This over here is a chromosome only when it's been doubled. Chromosomes don't typically look like this. They instead look more like this instead after they've been separated, right? But this central part here, what happens is this is the central part from which the chromosome is pulled into the two new cells that the cell is dividing into. Okay, so what we'll first discuss is just what mitosis is. But even before that, just because it's important, I want this write down for you that chromosomes are the compact or condensed store of DNA. Okay, and they come from the Latin word for chromo and some, where chromo means color and some means body. So chromosome, this means colored body, right? So you can see that biologists are very creative with names. All they do is they sneakily change the names to Latin instead. Okay, so with mitosis then, right? Mitosis is the type of cell division that where you typically have one cell, which originally will have n amounts of chromosome. Okay, in fact, we can actually call this 2n. So this is a cell that has 2n amount of chromosomes, where n is the chromosome number. So for example, for humans, the number of chromosomes that we have as pairs is 23. So every single cell of my body would be having 23 pairs of chromosomes, okay? So in total, we've actually got 46 chromosomes in us. But they exist in pairs because one of the pairs come from the mother and the other pair comes from the father, okay? So that is how it works in biology. And when I'm talking about the mother and the father, I'm talking about the parents with chromosomes XX and XY, typically speaking, at least. Okay, so with humans having 23 pairs, that means that we would totally be having 2 times 23 chromosomes in us right now, which is a total of 46. Okay, what happens is before mitosis can start, the cell first needs to grow. Okay, it needs to grow and develop itself so that it has a larger amount of cytoplasm, and it also has double the amount of organelles. For example, it might be needing double the amount of mitochondria before cell division happens. Okay, and the next thing that's going to happen is right before mitosis starts, the chromosomes are going to start to duplicate. So DNA gets replicated and chromosomes get duplicated. So that is going to happen right here, and we're going to see now it's 2 times 2n. So we now have basically 2 lots of 2 times 23. Okay, that's what's happening right now for the chromosomes. And then what's going to happen is the cell is now going to split up those chromosomes so that each cell that it divides into will end up having exactly 2n and also 2n. Okay, so each cell has the same genetic content. The key features of this is that the organelles are evenly distributed. The chromosome content is the same, or I can say that they are genetically identical, meaning that the end products are just clones. And another point as well is that 2 daughter cells are produced. Okay, so these are some of the key points of mitosis. And the main reason why mitosis occurs, there are 3 functions of it. The first function is that it could be because it is trying to grow the body. The second function could be that it is trying to repair the body, right? There is another one as well, but these are the main 2 reasons why mitosis would occur, right? It's for growth and repair of the body. So if you have a cut, the reason why that cut fixes is because of the mitotic cell division, right? If you are growing from a child to a teenager to an adult, that growth process will also be undergoing mitosis, right? That leads us to the concept of something that's called a mitotic index, where um, that's a quantity that's measured based on the proportion of the amount of cells that are dividing. So, for example, if you look at the number of cells of someone with puberty and you compare that to the number of cells of someone who's an adult, if you compare their mitotic indexes, what you would expect to see is you'd expect to see the person who's undergoing puberty to be having a higher mitotic index than the adult who is no longer undergoing puberty, because the adult is no longer growing in size, whereas the person who's undergoing puberty would still be growing in size, right? And the time of the life where the mitotic index is the highest is when someone is still an infant, because infants grow incredibly quickly for if you compare them to the other parts of life that they are part of, right? But this is essentially what is happening in mitosis, okay? So all of the organelles that are in the cells, like ribosomes and the Golgi bodies and so on, that all needs to be duplicated, okay? The other point as well is just that mitosis on its own forms a very small part of cell division. The majority of the cell division process happens when we aren't actually looking at it, right? That is to say that the majority of the cell division process happens in the preparation part, where the cell is still preparing and actually growing itself to be able to divide, right? So just something to keep in mind, that typically when you look at cells under the microscope, it doesn't look like they're dividing because they might be preparing for that cell division. So you're only going to see a few amount of cells that are dividing, and you'd be lucky to see the cells which are in the middle of cell division. It's quite rare to see them in that stage because, again, that's a smaller chunk of the overall cell cycle. Okay, so we've got different stages in the cell cycle. For example, if I represent it as a pie chart, the actual mitosis part is quite small, and the growth part over here would be even longer. And this part would just be the lagging part where it's not doing anything, but you'd have the growth part over here. This part is the S phase, which is the synthesis phase, where it's actually synthesizing different things, where it's, for example, duplicating the proteins and duplicating the DNA and so on. And then again, the actual mitosis part happens here. Okay, so the key point is that, again, it produces clones, which is what we need in certain situations. But that's not always going to be the case, right? And that leads us to what I wanted to mention as the third function, which is also in reproduction, right? So this doesn't really happen for humans, but this happens in asexual reproduction. Right? So asexual reproduction is when an organism is dividing with the absence of any other partners. So they're just dividing on their own. So that would be with only just one parent being present, and it produces offspring that are complete clones. So some organisms do this, for example, and we'll look into that more later on. But in asexual reproduction, mitosis is the only kind of cell division that happens. The other kind of cell division, which I'm going to go through now, that happens in sexual reproduction, which is meiosis. Okay, so again, this primarily happens in sexual reproduction for any kinds of organisms. Okay, in meiosis, starting off with this cell over here, we still have the 2N cell, right? But what then happens is the 2N cell duplicates the DNA, so we now have 2 times 2N, right? And for humans, let's write it down. So right now we have 2 times 23 for humans, and then we have 2 pairs, or 2 lots, I guess, of the duplicated 2 pairs of 23. And what will then happen is we have the first meiotic cell division that occurs. So this is the first cell division, which will produce 2 lots of the cells here and here, okay? And then finally, it will produce in another division over here, it produces cells with half the amount of chromosomes. Okay, so these cells have a special name. The cells that are having the original amount of chromosomes in the body and the pairs of them, you call them diploid cells, right? Di for 2, ploid for body again. So diploid means 2 pairs. And these guys over here, we call them haploid. Okay, and these in this case can be referred to as the sex cells or the gametes. So in the case of humans, that would be things like the sperm and the ova, or the ovum, which is the egg. Or in the case of plant cells, that could also be things like the ovules and the pollen. And also in the case of mushrooms, for example, that could be things like spores, right? So spores originally are actually just gametes that are made from mushrooms or fungi. Okay, but this is what the meiotic cell division essentially looks like. And the main advantage of, or the main reason why meiosis happens is to reduce the chromosomal number to a half, so that half of those chromosomes can fuse with the other parent. And the reason why this is done is, again, for sexual reproduction. It's to allow variation in the organisms, because now that the genes are fusing with another organism, like another mate, essentially, that will lead to variation in the organisms, and that prevents clones from being made. So there are some environments where making clones is not really that advantageous to the organism, in which case the organism will shift towards meiotic reproduction instead. But some organisms, for example, are locked, right? Some organisms can only produce or can only reproduce with mitosis, which is just cloning, and some organisms can only reproduce with meiosis, which is sexual reproduction, which is the case for humans, for example, right? And the main point here is, again, none of the cells now are clones. They all show genetic diversity or genetic variation because of the difference in the chromosomes between all of the cells. Okay, now that leads us to another point as well, which is just what stem cells are. So we discussed this briefly last time when we were talking about totipotency, pluripotency, and so on. Okay, but just getting back into it again, what you should be aware of is that stem cells, which is the germline original cells that make us up, which in the case of us would be the zygote cells or the embryo cells, right? So we all started from a single cell that was fused together by 2 haploid cells, right? When those 2 haploid cells fused together, they made this diploid cell. And that is the zygote which made us, and it was a stem cell because it had the capability of dividing into every single organ which we now have. Okay, that's what a stem cell actually is. And the way that a stem cell does this is through that differentiation process, which, like we discussed earlier, happens through mitosis. Okay, so mitosis will just differentiate all of the cells. into different kinds of specialized cells. Okay, now apart from this as well, remember that this takes place mainly in human embryos, or these cells are found in human embryos as well as in the zygote. Originally, that counts as a stem cell, and then it is also found in the meristem of plants. In case you're wondering what a meristem is, a meristem is the upper part of the shoot of a plant. So if you've got the plant where you've got the leaves growing here and so on, the tip part of the plant, which is growing the most in the shoot, that is what the meristem is. Yeah. So that part of the plant, that tip part is the part of the plant that is growing the most, and it extends outwards. The cells elongate over time and they start growing. Same way for the tips of the roots, those are the growing part of the plant. So there are certain species of plants where if you just cut the top part of the plant will actually no longer grow properly. So those are usually for the simpler versions of plants. For the more complicated species, that doesn't tend to happen. Talking about these stem cells as well, there's a lot of potential with them, but also a lot of controversy involved. With stem cells, the benefit is that since any one of these cells can develop and produce organs, that basically means that stem cells could potentially be used, and currently are, be used to grow organs or different parts of the body to treat diseases. Okay, so for example, if someone had a liver failure, you would theoretically be able to take that person's stem cells and then grow that stem cell into just the liver, take that liver and then use it as a liver transplant for that person. So there's a lot of potential here, but there's also a lot of controversy because there are ethical arguments against it as well as religious arguments against it. Now, again, because this is just science, when I'm not talking about whether something is right or wrong, right? That's to do with morals, which I'm not doing. But just to make you aware, there is controversy regarding this area where people have disagreements on whether it should be done or not. But just the science behind it is that this could potentially be used to treat people. The ethical issue behind it, though, is just the fact that it could lead to a slippery slope case again, where basically if you are allowing people to grow livers as organs, then what is the point beyond which we no longer allow it, right? It could maybe, in a very strange kind of world, lead to people growing humans in tubes and so on, right? That's like in a sci-fi, kind of dystopia kind of world where you're using stem cells in this kind of way. But that is something that could potentially be done. Either way, though, this could be used to treat things like liver failure. It could be used to treat things like diabetics and so on. Okay. Another thing as well is that with mitosis that we were talking about earlier, or with stem cells, right? We could also be using them. So this underneath section, so this talking about cloning with stem cells, right? We could potentially use this to revive extinct species. Because really, all you need is one cell from that species. You take that cell and you induce it under the right amount of electrical signals and the right amount of chemicals, and that could potentially make the cell a stem cell again. If we do achieve that, then that cell could keep on replicating and duplicating under mitosis to produce the organism again, right? So if you have extinct species or if you have rare species that are endangered, those could be revived again. And we've done this for some animals, and we've also done this for some plants. So that's something to consider as well when we're talking about stem cells. Now, apart from that, all of that just comes under a brief glance of mitosis and meiosis. We'll be looking more into that later on when we're covering reproduction. But just beyond that, you should also be aware of the fundamental processes that are happening in cells, which I alluded to earlier in the beginning of this video. But there are 3 processes in organisms in their cells that you need to typically know about. for biology, okay? And these are really important because they're fundamental in the sense of you will see them not just now, you will see them being mentioned over and over and over again. So it's important that you keep these in mind throughout your learning process for biology, okay? So we'll start with the simplest one, which is diffusion. Okay, and here is the formal definition of diffusion. We say that diffusion is the net movement of molecules or chemicals from a region or from their region of higher concentration to their region of lower concentration. Okay, there are other definitions of this as well. My favorite one is just saying that it's the net movement of molecules down a concentration gradient. That's just fancy talk for the fact that the molecules are going from a high concentration spot to a low concentration spot. Okay, so just even without biology, just explaining this without biology for a moment, right? If you have a beaker of water and you put in a tea bag over here, or let's say some salt, what's going to happen is that salt packet is not going to stay over here. It's eventually going to spread across the beaker. Okay, so it goes from a high concentration here to its regions of low concentrations until equilibrium and stability is reached. Okay, so the reason why diffusion occurs is because every particle normally moves with some amount of kinetic energy where it's just moving around, right? So this links back to physics when we were talking about kinetic energy and heat energy, right? Every particle is moving randomly on its own. And when those particles move randomly on its own, they have a higher chance, if they're all concentrated in one area, they have a higher chance of spreading out than they have ever a chance of coalescing back. That's not to say that molecules can't coalesce back theoretically into this one single spot, but the likelihood of that happening is just so slim that when we're talking about it energetically, right, it's way likelier that the molecules that are at one spot all will spread out and then dissipate equally throughout that solution. Okay, whether it's sugar or whether it's salt or whether it's some kind of chemical or food dye that you're putting into that water solution, it will spread out. And this doesn't apply only for water, it also applies for gases as well. So for example, if I have the gas around me right now, if someone in the room that's with me opens up a potato crisp packet, I guess, right? What's going to happen is the scent of those chips are going to float in the air. When they float in the air, I'm going to smell it from where I'm sitting. And that scent dissipates around the room through diffusion because inside the potato chip packet, right, all of the chips are having a high concentration in their scent, and then as that scent travels out, it goes throughout the room, and now the room is imparted with that equal concentration across all sides of it, I suppose, with the scent of the potato packets, right? So again, that's what's happening in diffusion. Another point as well, just to mention, is you should be aware of what are the factors that influence diffusion. Okay, that firstly, it's influenced by the speed of the molecules, which is to say the temperature of the solution, because the temperature decides the kinetic energy and the speeds of all the particles. So if the temperature is very quick, then the diffusion rate is going to be even quicker. And another thing that it depends on as well is the concentration gradient. That is to say that if the concentration is higher, then the rate of diffusion is going to be quicker. Okay, and then it also depends on one more thing, which is the SAVR. What is that, you might be asking? SAVR stands for the surface area to volume ratio. So if you have a beaker, so just to explain this point here, if you have a beaker where you have dropped a salt pellet into it that's a cube, this will have a harder time dissolving as compared to another beaker where you have a salt that is now placed in a crumble like this, or if I had to increase the surface area, maybe I can do something like this. If I put a salt in this kind of weird shape here, this would have more area that's in contact with the water molecules to pull apart all of the salt molecules here, right? So you can see the surface area to volume ratio is higher. So they both might have the same volume, but because the surface area of this salt packet is higher, the salt packet can be attacked more easily in this solution, I suppose, right? So again, that's what's happening in this between both of them, that here there's a higher surface area to volume ratio, so this will have an easier time dissolving. Okay, this relates to something that's called Fick's law. Okay, so it's just something, it's a bit of a shorthand for you to keep in mind, but Fick's law basically states that the diffusion rate is proportional to the temperature times the concentration gradient all over the surface area. Oh, sorry, that's also included here. Surface area to volume ratio. Now, there's one more point as well that the diffusion rate is impacted by negatively instead, and that is the diffusion pathway. Okay, so to explain what that means, if I have a beaker here, and let's say that I have two, like I have a solution here fully, but I have a box here with paper around it, and another box here with paper around it. And inside here I have, let's say, blue dye, and over here I have, like, let me make it red. So I have red dye here, okay? These papers over here are going to influence how easy it is for the red to travel into the blue and vice versa, right? That is to say, if the paper is thicker and larger, right, it's going to decrease the rate of diffusion. It's going to make it harder for the blue to travel to the red. If the paper is very thin, then it's going to make it easier for the liquids to actually exchange. And just be mindful as well that the reason why we're discussing about this is because this is an essential process that happens throughout organisms, right? And I can think of countless places where it happens in biology. So as one example, right now, as I'm breathing in and out, the way that the lungs is exchanging oxygen and carbon dioxide with the cells of my body is through diffusion. So I've got some gases in my lung. Those gases that are in the lungs will actually diffuse across to the blood vessels, right? That happens through diffusion. Another example is just when you are eating food. If you are eating food, then what's going to happen is the sugar that's in the intestines after you're eating the food is a high concentration. And if you are starving, then the blood has a low concentration. So the sugar will go into the blood through that diffusion process as well. Okay. So everywhere throughout the body, this diffusion process happens. It's even more important for primitive unicellular, like single-celled organisms, right? Because for single-cell organisms, the way that they exchange materials with their surroundings can be through diffusion, okay? It's a bit more complicated in reality, okay? It's not just as simple as diffusion. There are different kinds of diffusions. There's also different kinds of transports. In some cases, you have co-transport, but we'll go into that some other time. But the main point for now is you need to be aware that the rate of diffusion is actually dependent on these four factors over here. Okay, and another point as well, just linking this to physics, is diffusion is a passive process, right? What that means is that it happens spontaneously and naturally. Okay, meaning that it does not require an energy input. Okay, a more complicated way to say it is that this is thermodynamically favorable. It's energetically favorable, right? So you don't need to put in any process. This will always happen naturally whether you like it or not. Okay, so in some cases it's a boon for organisms, but in other cases it's also a hindrance because some organisms will be fighting against this. One example I can think of for this is that if you've got a cell that is trying to store food in the body, that cell has to actively use energy to keep that food locked in place, to prevent the body from digesting and spreading out that food to the rest of the body, because it needs to be kept in this one area, right? So that's an example where essentially the organism is fighting back against the passive process of nature, right? But this over here is just what diffusion is, okay? And again, you see this happening in solids and liquids as well. I mean, liquids and gases, right? Now, apart from diffusion, the other process as well that you should know about is this osmosis. Osmosis is a process that happens typically with reference to water, right? So the definition of osmosis is just that it is the movement of water molecules from a region of higher concentration. And I'm talking about higher concentration, I'm referring to higher concentration of the water itself. I'm not talking about the salt concentration, okay? So just to give you an example of what I mean here, if you have a solution that has a high amount of salt concentration and you have a solution that has a low amount of salt concentration, then inversely, the low concentration of salt solution means that it has a high amount of water, and the high concentration of salt solution means that it has a low amount of water. Okay, so just be mindful when I'm talking about concentration here, I'm actually referring to not the salts that are present, but I'm referring to the water itself. I'm talking about the concentration of the water. Some people don't like this confusion, so rather than talking about concentration as a word, they instead use the word water potential. Okay, so water potential refers to how much water is in that substance. So for example, instead of saying high concentration, I can also say high water potential. Okay, so it's a movement of water from a region of high concentration to a region of low concentration, or you could also use the word lower water potential. And the key point here that makes it different to the previous one is that it is through a semi-permeable membrane. Or you could also say partially permeable. Okay, there's a slight difference between them. A partially permeable membrane is one that has no autonomy over what it's choosing to enter and what can't enter. So it has fixed rules on what can enter and what can't enter. But a semi-permeable membrane is a membrane that can kind of modify its rules every now and then. So you see that a lot of cell membranes in biology are actually semi-permeable in this way. Okay, but back to osmosis, right? So just to give you an example of what I mean. Okay, and I think a really good example for this is with blood cells, and I'm talking about our red blood cells that we have in our blood. Okay, if I have a beaker of, let's say, pure water, right now this beaker has a high potential of water. Okay, because it is just pure distilled water. Now, if I take one of my red blood cells and I put them into this beaker here, of course I'm exaggerating the size of a red blood cell. Of course it's going to be way smaller than this, but this to show you what happens. In this red blood cell, naturally in my body, okay, this actually has salt present in it. And because it has salt present, it now has a low potential of water. So this has a lower amount of water. If you're trying to understand what actually is going on here, right, what happens is because it has a lot of salt ions present, those ions which are charged will attract the water. So the water starts to flow in and it stays there because it is attracted towards this this cell over here, okay? And just highlighting another misconception really quickly, okay? Just be mindful that in both osmosis and diffusion, the movement doesn't only occur one way. There's always movement between both of the areas. That is to say that there's water flowing into the cell and there's also water flowing out of the cell. But in osmosis and diffusion, we are talking about the net movement. Okay, that there's an overall net movement, an overall like total movement from one area to another, right? So in this case, what's happening is because there's a lot of salt present in the cell sucks up all of that water like a sponge, right? So there's going to be a higher amount of water entering the cell than there is water leaving it. And eventually, what's going to end up happening is, over a lot of time, that cell is going to fill up and fill up and fill up to the point where it actually bursts, right? And this is what happens to animal cells. If you place them into pure solution, the pure solution floods into the cell takes in all of that pure water, and it will burst as a result, right? So that is a result of the osmotic pressure that builds up inside the cell. And this osmotic pressure, the same pressure that you're seeing over here that's killing an animal cell, that can also be the same kind of pressure that actually saves a plant cell. Because in a plant cells actually have a cell wall around them, right? And the benefit of having a cell wall around it is that that cell wall is rigid and is able to withstand the osmotic pressure from water going into the plant. This is the reason why if you water your plants, they will no longer be wilted and they will instead be standing upright and rigid. That's because of the water that's flowing in, and it's held in place by the scaffolding of the cell wall that's around it, right? So you can see an example there of osmosis being really important in biology, right? So important to the point that basically if someone doesn't have water for a day, they wouldn't be able to survive that well. And I think the time limit is like a week. After a week, a person will die after not having water. And for food, it's around a month, I believe. That's like this when starvation sets in. I might be wrong with these facts, and you can always check them. But the main point that I'm trying to just emphasize is that water is incredibly important to all of life, not just us. And that's because of us needing to maintain these different kinds of osmotic potentials in the body, right? And there are other reasons as well, like the fact that water is needed to dissolve salts, and we need those salts for different functions in the body. Okay? But again, that's just an example, right? And in this case as well, the semi-permeable membrane is the cell membrane. But this doesn't need to happen only in things that are alive, like red blood cells. They can also happen in things that are not alive. For example, you could look at a whisking tube. I believe that's what it's called. This is the same kind of tube that people wrap their sausages in. So if you've got a sausage and you are wrapping that sausage, right? The material that sausage is made up of is this exact same whisking tube. And if you zoom in on that material, it's got a lot of these tiny holes in there, which is only small enough to allow water to pass through, to permit water to pass through. Okay, so if you've got a packet of some whisking tube, right, and you have a lot of salt in that whisking tube, and again you put this into a beaker, what's going to end up happening is that the whisking tube is going to start expanding. Okay, so again, the tube expands due to endosmosis. And endosmosis just means that the water is going into the solution, right? So it's inflating. So endosmosis, inflating the tubing, right? So that's an example of where osmosis is happening, not with something that's alive, but with something that is just dead, I suppose, right? Because you normally harvest this from the animals usually. But again, that's what's happening in osmosis. And just like diffusion, osmosis is also influenced by the same factors. That is influenced by the concentration gradient, it's also influenced by the temperature, and it's also influenced by the that pathway that's present between them. Right? So these are the different factors that will influence osmosis. Okay? So endosmosis is when water is going into a substance, and exosmosis is when water is leaving a substance. So similarly, if I have a red blood cell, and I put it into a very salty solution, okay, much saltier than the body, okay, then what's going to happen is that the water from this red blood cell is slowly going to start leaving the cell in exosmosis. So the cell is going to start shriveling up. Right? If I have the same kind of beaker, the cell will start shriveling up as a result. Okay, and you can see this very easily as symptoms of dehydration. If someone is dehydrated, then what's going to happen is that their skin is going to have a lot more cracks in it, a lot more like a lot more shriveling present because of the lack of water, right? And that again shows you how important water is, right? So some organs as well, I believe, are made up, like majority of some organs in the body are just water as well. And that again goes to show you how important water is in keeping our structure afloat. Okay, but there's this osmosis, and again, like diffusion, it is also a passive process, or a kind of passive transport, right? Now, both of these osmosis and diffusion are both contrasted to active transport. Because if you think about it logically, okay, we need a way of storing substances into one location that is against a concentration gradient, because that's really what's happening in digestion. When we eat food, right, we are taking in an excess of the nutrients and we are storing that nutrients inside our body. We are preventing those nutrients from leaving our body. The way that that happens is through a process that's called active transport. Okay, and here is the typical definition of active transport. Now, active transport is the movement of a substance, or the net movement if you want, right? So the movement of a chemical up a concentration gradient. So rather than it going down the concentration gradient, it is going up a concentration gradient. If you're wondering what the word gradient means, gradient just means the gradual change, right? So if you are going from a high area to a low area, that's a gradient of change. It's not only used in this context, it can also be used in art as well. For example, in art, you might be referring to color gradients to show how the color is changing over time in some kind of art project, right? But you use gradient all over the place. Anyways, just continuing. So it's a movement of a chemical up a concentration gradient with the use of energy. Okay, so again, this is an active process. So for osmosis, as well as diffusion, the organism doesn't need to spend any of its energy because these are natural processes that occur whether you give it energy or not. But with active transport, the organism does need to spend energy making this process happen, right? And usually the way that it does that is by using the energy that's made by the mitochondria, which is ATP. So this ATP over here will bind to these tiny little carrier proteins, and those carrier proteins will flip in just the right way to move substances across a membrane, right? So again, if you've got a cell membrane, and if this cell for some reason needs a lot of sodium inside and a lot of potassium outside, what the cell might do is it might move a lot of sodium inside through active transport, and it will move a lot of potassium outside the cell through active transport again. Okay, so that's just a bit of an example. And this process of what I've just shown you over here is a simplified explanation of what's actually going on in a nerve cell. Okay, but that's not the only place where you see active transport happening. You would also see active transport happening in, let's say, a root hair cell of a plant, which looks typically like this. Okay, with the cell wall around it. So this root hair cell is going to be having mitochondria over here, a bunch of the mitochondria. It's not going to have any chloroplast because obviously the roots of the plant are not going to get any sunlight because they're underground, so it's not going to have any chloroplasts. But it is only going to be having mitochondria in it. That mitochondria will generate the ATP, which is the energy, the chemical energy, that is needed for active transport to occur. So in this case, right, what the plant wants is from the soil that's around it, it wants to uptake minerals and ions inside. Okay. Osmosis, on the other hand, will be taken, or rather, water, on the other hand, will just be taken through osmosis, right? You cannot really active transport water in a sense. It's a bit complicated. So many organisms just need to rely on osmosis to take in water. The only other way that you can indirectly take water is by making yourself really salty. Okay, and just to explain what I mean by that, if an organism has a lot of salt in them, the salt will magnetically, or I guess I should say electrically, pull the water towards it, right? So in this case over here, what's happening is this root hair cell is going to uptake a lot of mineral ions by active transport. That will produce a higher concentration of salts inside the root hair cell. As a result, any water that's on the outside will get pulled into this root hair cell by endosmosis like a sponge because there is a lower concentration of water inside, right? So that essentially is what's happening to pull up the water into the root hair cell, okay? But in this example for active transport, this is happening with the mitochondria releasing that chemical energy needed for this to happen on the cell membrane surface, okay? So all across the cell membrane, that is where active transport actually happens. But on the cell wall just provides a protective rigid barrier. Okay, so cell wall doesn't take place in any kind of active transport process. It's just cell membranes. That's the most important. Now, Another point as well is like how diffusion as well as osmosis were affected by different factors, so is active transport. As you can see in the case of this root hair cell, it has an appendage of sort that is long and extended outwards, right? So this appendage has a low volume, but it has a higher surface area, okay? So that's the hair cell part of that root hair cell, okay? And what that does is it increases the surface area to volume ratio, which increases the amount of contact area that's that's there with the soil, and that allows it to absorb more of the nutrients from that soil essentially. Okay, so again, just going back to this over here. So keep in mind that this rate over here applies not just for osmosis, it also applies for what do you call it? The active transport rate as well. Okay. And the other place as well where this happens for animals is in the digestive system. So if we've got a digestive system where our small intestine maybe looks something like this, okay, if I zoom in to one part of the small intestine, okay, we have the intestinal lining here. And next to this intestinal lining, we will also have a capillary here. A capillary is a very tiny blood vessel, right? So we'll have blood vessels here, with this blood vessel here, which is having red blood cells in it, and we'll also have a lot of, let's say, sugar, salt, etc. that we've eaten from the food here, amino acids as well. Okay, so what's going to happen is once we've eaten this in our food and once it's fully broken down to these components, now it's actually small enough to pass the cell membranes. So these cell membranes will have special protein on them, where these protein will use energy from the cells that are over here. So again, we've got a cell here, we've got a cell here, we've got a cell here, and so on. Okay, so each of these cells will have their mitochondria, which will give the proteins energy, and then these proteins here will carry the sugar towards the blood vessel. So again, the salt towards the blood vessel and the amino acid towards the blood vessel. So all of these sugars, all of these salts are then moving towards this blood vessel and they are accumulating over there. So regardless of whether the blood vessel has a high concentration of sugars or whether it has low concentration of sugars, this process of active transport is going to happen, right? So what that basically means is if you have a lot of sugar in your blood and you eat food despite that, that is just going to increase the sugar levels even more, right? Now, hopefully what's going to happen is that hopefully the body will compact that sugar to decrease the blood sugar levels. But, even in cases where the body is not working as intended or working normally, then this process will keep on happening regardless. The body doesn't, for digestion at least, the body doesn't have much leeway over controlling it. And again, it requires energy because it is building up chemicals. Okay. On a deeper level of understanding, the reason why this requires energy is because this is essentially going against nature, right? When I'm talking about going against nature, I'm talking about processes that are thermodynamically favorable or not. All right. So remember that when we're talking about it in physics, everything needs the lowest amount of energy in order to remain stable. Okay. So really, we're just talking about stability over here. The most stable system is a system where everything is evenly spread out because that will have the lowest amount of energy. Okay, the analogy I can think of for this is you think of it like if you have a lot of sugar that's compacted into one area, there is a lot of energy stored in that sugar. So what that sugar molecule would really like is to be able to spread out, to distribute that energy evenly. Okay, so that is a difference of a region of a low stability, which is when it's all compacted, to a region of a higher stability, which is when it's all compressed, or rather expanded outwards. Okay, so for diffusion and osmosis, you've got molecules that are flowing from a region of, let's say, a high concentration to a region of low concentration, because that is dispersing energy. Okay, but in active transport, you're going against that cycle. So you have to spend energy trying to compact everything together to make it more, I guess, unstable, right? So that's where that energy is needed. And usually what will happen is that energy will eventually be dissipated away as heat energy to the surroundings, right? All of this is to do with entropy as well, right? Which is a concept where if you have a system that's very chaotic, that has a high entropy, and if you have a system that is less entropic, then that has a lower, like, I guess it's got more order to it, it's got low chaos in it, and so on. Okay, so as an example, if your liver has successfully taken all the sugar and it's compacted that sugar into glycogen with it storing in it, like it's storing it within the liver itself, right? That would be having low entropy. So that means that in order for the liver to do that, the liver actually needs to use up energy to make itself have low entropy. And that is really what life is about, right? So again, life is about using using energy to maintain its state of low entropy, right? And we will perpetually continue doing this until the aging process gets the better of us. And meanwhile, in diffusion and osmosis, not we don't need to spend any energy on that because we are just following the universe's natural flow of entropy, right? Where you will have the organism that doesn't need to spend any energy because that's just what things naturally do. Okay, so everything in the universe will always tend towards a system of this higher entropy over time. That's just how it works, right? And if you're wondering what the formal definition of entropy is, you can think of this as the maximum number of possible configurations of a system. Okay, but the analogy that you can think of it as is just that if you got something that's really messy, then it's very entropic. If you got something that's very clean and organized and neat, then it's less entropic, right? So that is the reason why diffusion and osmosis are passive transport processes, whereas active transport is an active process that's for which we need energy. Okay, that also means that if you've got a cell and you see a lot of mitochondria in that cell, chances are that cell is involved either in movement, or if not in movement, then that cell is involved in active transport, right? So all of the cells in the small intestines, those would be having ample amounts of mitochondria, and they would be having high amounts of mitochondria because they need to actively move out the digested food in the small intestines into the body. Okay, and it's not just the small intestines, it can also be, for example, the kidneys, right? So in the kidneys, right, there are particular cells where if you've got the blood vessels in the body, so I'm just going to call this the blood, and you've got the, let's say, the nephrons of the kidneys, so the kidney cells. And let's say that this part over here is, for example, the actual urine that is produced by the kidneys, okay? What's going to happen here is if we have too many salts in the body, or if we have any toxins, let's say like a poison, okay, what the body is going to try and do is the body is going to use active transport to move this process, or to move the poisons towards the kidney cells, and then from the kidney cells into the urine. Okay, that's typically how the body gets rid of poisons. It's a bit more complicated than that. There's actually a lot of pressure involved and so on. But the point either way is that even with that higher pressure involved, that higher pressure is just another form of energy that the body is using to move the poisons from one place to another. Okay, and when I'm talking about poisons, it doesn't necessarily just mean like the snake venom and stuff like that. It can also mean stuff like waste materials, like urea. So urea is the most deadly one because urea is produced as the byproduct of protein waste. So typically, if someone eats a lot of protein and then that if we have an excess protein that we don't need anymore, then the body will break down that protein, and as part of that breakdown process, it produces urea, and urea is what is very harmful to the body. So the body will take that urea and it'll move that urea towards the from the blood through the kidneys into the urine, and that's where it actually will leave the body, right? But that again happens through active transport, not through anything else. Like overall, it would happen through active transport. And again, once we study a bit more about the kidney, you will learn about this in more depth. But for example, if there was something in the urine, like sugar, say, that the body didn't want to get rid of, then the body would reabsorb that sugar that's in the urine through active transport again. Okay, so again, if the body mistakenly removes any kind of sugar, it will reabsorb it through active transport. which seems like a flawed system, but again, it'll make more sense once you study a bit more about the kidneys. Okay, but otherwise, that essentially covers what is the basics of mitosis, meiosis, stem cells, and the three vital processes of diffusion, active transport, and osmosis. Okay, so for the next session, what we'll be looking at in biology is we'll be focusing more about what are the different organizations of life. So that is to say how life actually is divided on a smaller unicellular scale all the way to the more complicated levels like organ systems and so on. So that's quite interesting. And otherwise, for the next video that I will upload, we will go back to chemistry. So again, I hope you enjoyed this particular session. And if you did enjoy, please feel free to support me on Patreon. If you have any questions or topics you want me to cover, please do comment in the comments below, as well as any criticism or feedback will be appreciated as well. Right. Thank you, and see you next time then.